As Dr. Delfino said, my project is on the prevalence of E. coli and fecal coliforms on equestrian workers' boots throughout central Kentucky. Just some background information. Escherichia coli, or more commonly known as E. coli, is a bacteria that is typically found as normal microbial flora within human and animal intestines. Humans can be infected through contaminated food, water, or contact with people or animals. Fecal coliforms are used as indicators of contamination in water or on surfaces. Both E. coli and fecal coliforms can be harmful to human health if ingested. A study done by Chambers and others in 2006 found E. coli and fecal coliforms present on both ATV tires and boots in Alaska. This study supports that E. coli and fecal coliforms could be carried into the communities. Another study done by Snedeker and others in 2009 compiled data of E. coli infection cases and found that about 19% were caused by secondary transmission. The feces of farm animals provides a primary route for human infection by E. coli, as a study done by Duncan and others in 2000. A study done by Shasta and others in 2012 found that the most prevalent time for E. coli to be passed through the horse species was during the months of August through October. The purpose of this study was to examine the prevalence of fecal coliforms and E. coli on equestrian workers' boots throughout central Kentucky. My first null hypothesis is that fecal coliforms will not be found on the boots of equestrian workers in central Kentucky. My first alternative hypothesis is that fecal coliforms will be found on the boots of equestrian workers in central Kentucky. My second null hypothesis is that E. coli will not be found on the boots of equestrian workers in central Kentucky. My second alternative hypothesis, that E. coli will be found on the boots of equestrian workers in central Kentucky. All of the samples were collected in the morning, averaging between 8 and 11 a.m. We collected them during the fall of 2012 from 4 different horse farms. Each sample was taken from each worker's right boot, sole, and sides. They were collected using sterile swabs and were then placed immediately into Stewart's transport medium. They were then taken to the lab within 2 hours of collection. The samples were then streaked for isolation on Colascan Easy Gel plates under a laminar airflow hood. Then they were placed in an incubator at 35 degrees Celsius, and at 24 and 48 hours, they were read and counted for the presence or absence of E. coli and fecal coliforms. The upper right-hand picture is a picture of my control plate. The pinky red side is the fecal coliforms. The dark blue purple coliforms is E. coli. And then underneath is Staph aureus. We plated up to show that it wouldn't grow on these plates. The upper left-hand picture is the incubator where the plates were placed. And then the bottom picture is my workstation under the laminar airflow. Again, we collected from 4 different sites. We collected a total of 45 samples, with 24 or 53.3% positive for E. coli. 21 or 46.7% were negative for E. coli. 42 or 93.3% were positive for fecal coliforms, and 3 or 6.7% were negative for fecal coliforms. 2 or 4.4% were negative for both E. coli and fecal coliforms. From Farm 1, we collected a total of 26 samples. 12 or 46.2% were positive for E. coli. 14 or 53.8% were negative for E. coli. 23 or 88.5% were positive for fecal coliforms, while 3 or 11.5% were negative for fecal coliforms. 2 or 7.7% were negative for E. coli and fecal coliforms. From Farm 2, I collected 13 samples. 7 or 53.8% were positive for E. coli, while 6 or 46.2% were negative for E. coli. 13 or 100% were positive for fecal coliforms, with 0 negative for fecal coliforms and 0 were negative for both E. coli and fecal coliforms. From Farm 3, I collected 4 samples. 3 or 75% were positive for E. coli, with 1 or 25% negative for E. coli. 4, or 100%, were positive for fecal coliforms, and 0 were negative for fecal coliforms. Also, 0 were negative for both E. coli and fecal coliforms. From Farm 4, I collected 2 samples. 2, or 100%, were positive for E. coli. 0, or 0%, were negative for E. coli. 2, or 100%, were positive for fecal coliforms, while 0 were negative for fecal coliforms, and 0 were negative for both E. coli and fecal coliforms. This is a composite table of the positive and negative results for both E. coli and fecal coliforms. And this table just has it broken down between each farm of the positive and negative results for E. coli and fecal coliforms. The right-hand side are pictures of my results. As you can see, the colonies are very numerous. The pinky red colonies are the fecal coliform ones, and the dark purple, dark blue colored ones are the E. coli colonies. And on the left-hand side is me working under the laminar airflow hood, streaking the plates. And now for my discussion. My results did not support the first null hypothesis that fecal coliforms will not be found on the boots of equestrian workers in central Kentucky. My results did support my first alternative hypothesis that fecal coliforms will be found on the boots of equestrian workers in central Kentucky. The results did not support my second null hypothesis that E. coli will not be found on the boots of equestrian workers in central Kentucky. They did support my second alternative hypothesis that E. coli will be found on the boots of equestrian workers in central Kentucky. Chambers' study with others in 2006 in the Alaskan community found E. coli and fecal coliforms on the boots. My results support this, that these bacteria could be carried into the communities. Snedeker and others in a study in 2009 again compiled data of E. coli infection cases and found that about 19% were caused by secondary transmission. Equestrian workers wearing their boots into the communities could be contributing to these secondary infections. This study had several limitations besides time and money. We had a small sample size and were only able to test a few locations. The boots also may have been worn to other farms and could potentially be carrying bacteria from those other farms. We had a small testing radius due to the fact that the samples needed to be in the lab and streaked within 2 hours of collection. Also, the health at each farm of the horses was unknown. For future studies, I would recommend to test farms outside of central Kentucky. Test at different times of the year, have a larger sample size, test the same numbers of samples at each farm, and compare those results. I would also like to conduct a survey to ask the health of the horses at each farm and whether the individual owner of each boot cleans his or her boots regularly or not, and also to test at more locations. I would like to thank Ms. Jewett, my research director, for all of her help, her guidance, and her support throughout this project, very long project. And of course, Dr. Delfino for his instruction, his guidance, and always pushing me to be the best that I could be in this project. Mr. Day for ordering my supplies and making sure I had everything and always asking about how my project was going. I appreciated that. And my friends and family, of course, supporting me through all the tears and the success of this project, and especially Mel through IVR. I don't think I would have made it without her. Any questions? Dr. Young?
Just curious about maybe some of the E. coli students and those that work at farms, if they should maybe like change their boots before they come to lab.
I would suggest that probably be a good idea. Any other questions?
Courtney, could you just comment a little on how that secondary transmission might occur, what we're talking about, some of the
Okay. Secondary transmission means that they did not pick up that bacteria from the direct source. A primary transmission would be that they were working directly with the animals or people, they picked up the bacteria then. Secondary means, somebody else who originally picked it up from the primary site, brought it into the community, and that person unknowingly picked up that bacteria. Okay, thank you.